Good evening.
Yeah, so
Now we switch on to English for everybody.
Okay.
It's my great pleasure to introduce Kshitij Mohan Singh, he is one of our Vidyapith brothers, and he is 3 years junior to me, I think. 3 years, right, Kshitij? You
No,
passed
I
out?
think more. I think 6 or 7. I passed in
Oh,
2007.
wow. Oh, right. You passed in 2010. Right. Yeah.
7.
7. That
Yeah.
makes it. Oh, you passed 2007 class 10. Right. Okay. That
Yeah.
makes around 5 years. Okay. Regardless, he's one of the most illustrious people from Ramakrishna Mission Vidyapith. And to introduce him briefly, he passed class 10 from Vidyapith in 2007. And after that, Kshitij goes to the Indian Institute of Science Education and Research in Mohali to do his bachelor's and master's in biological sciences, where he was also given the Academic Excellence Award for the best student in the batch. Subsequently, Kshitij moves to Tübingen for his PhD at the International Max Planck Research School, where he studies the changes in age-associated gene expression and epigenetic landscape in cerebral endothelial cells, which are basically associated with blood cells in our blood vessels in our brain. The context was to try to understand how brain hemorrhage is related to increasing age. And currently, Kshitij is a postdoctoral fellow at the University of Oxford at the Division of Cardiovascular Medicine. And again, he is studying epigenetic changes that develop during cardiovascular development in the context of trying to understand myocardial infarction, which I get to understand is technical words for induced heart attacks or heart attacks in general. So basically, what I am interested in, which gets me very curious about, is the word epigenetics, which, if I understand correct, is the study of how two siblings, even with similar gene sets, they tend to show different qualities. And why that happens is because of switching on and off of genes in certain contexts, which is what they call क्या होता है? There is a word called nature versus nurture. Probably this is also
Nature
what is
versus
related.
nurture.
This is
Yeah.
also what is related to it, I think. Yes. So these are things that Kshitij will tell us now in the next 1 hour. Over to you, Kshitij.
Thanks a lot. Thanks a lot for the lovely introduction, Pratyushda. Just starting with your basic question, like the nature versus nurture. So the nature is something that we basically inherit. Nature is the genes that we have, and we do not have any control on what we inherit. And nurture is the part which can be anything. It's not the nurture in just the literal sense of how your parents or how you are nurtured. It extends to almost everything. It extends to your lifestyle, your stress, your food, the chemicals that you are exposed to, everything. So that's what I think you are interested in, like what is nature versus nurture. And I will talk briefly about that part as well, like why, despite two identical twins having completely like 100% genetic similarity, why they are not completely identical in all aspects. So that will come later, but before that, I'll start with basics. So before starting with epigenetics, I will give you all a brief flavor of how exactly what is a cell, how genes function, how genes are present in our body, and everything, basically all the basics related to genes and cell and molecular biology, so that I can build on the foundation and then also give you a brief flavor of what is epigenetics. Yeah. So every life, every mammalian life specifically, and all the other lives as well, as you know, they start from just a single cell. So when the sperm and the egg fertilizes, it's a fertilized egg. It's just a single cell. So that single cell, it keeps on dividing. It divides into multiple cells, 1 into 2, 2 to 4. And ultimately, when it gives rise to an adult human being, they have almost 40 trillion cells. So 1 trillion is 10 with 12 zeros. So we have 40 trillion cells in our human body. And all of them, they start with just one cell. So if there is a one cell which is the starting material. Basically, all the 40 trillion cells have come from that single cell. And therefore, the genetic makeup of all the cells is exactly the same because you started with the same single cell. So every cell, every 40 trillion cell in your body has exactly the same genetic makeup. So as you can see, it starts with one cell, then goes to 2, 4, 8, then it forms. I'm not getting into details of what is morula, blastula. These are all developmental stages. And then this is a very important stage, which is called gastrulation. So gastrulation is when it's still like close to a few hundred cells at that time. But at that time, what happens is the pattern formation starts. So the cells have started to become specialized, and they have committed to a certain fate. Like these cells will become the inner part of the body, certain cells will become the outer part of the body. So there's not a complete commitment. So because you have more than 200 cells, 200 different types of cells. So there are 40 trillion cells and 200 different types of cells. So at that stage in gastrulation, not all the cells have not committed to all the 200 states, but they have some commitment from which there cannot be much reversal. So anyway, so what happens at this process? So the process from making 1 to 2 cells is called division, but how does a single cell, which is exactly the same starting point, how does it get divided into different kinds of cells? So that process is called differentiation, because your heart cells is different from the kidney cells, and your kidney cells is completely different from your skin cells. So how does that single cell form different types of cells? That process is called differentiation. This gives rise to a fully functional, extremely complex system, like any mammal that can breathe, eat, think, reproduce, basically do all the metabolic processes. And most importantly, it's a self-sustaining, it's a completely living being. So how does that happen? So as you can see, so in the center is the initial zygote, and that basically gives rise to hundreds of cells, more than 200 different types of cells. And as you can see, they are completely different in morphology as well as function. So for example, if I start with an immune cell, what is an immune cell? It basically, its function is to protect you from external enemies. So whenever there is a bacterial or a viral attack, it goes on to kill the external invaders. Similarly, you have epithelial cells, which is the outer lining of the skin or any of the lining. So what's its role? Its role is to protect you from, it basically gives a tight barrier and protects, forms a semi-permeable barrier. Then you have nerve cells. So what are these nerve cells? They are one of the very few unique cell subtypes in our body. They can generate electricity and conduct electricity. And that's how a signal from different parts of the brain to the body and body to the brain, or the entire reflex works. So when you are basically thinking, there is an electrical activity going in the brain. When you are hurt and you all of a sudden your hand goes towards the injured part, that reflex action is also due to electrical signal. The electrical signal from, say, if you injure your feet, the nervous system sends an electrical signal to your brain, and then the brain processes it and again sends you a signal back to your hand so that you pull the thorn or nurture your injury. So this nerve cell, it also started from the same cell, but it can conduct electricity. But the immune cell cannot conduct electricity, or epithelial cell cannot. Similarly, muscle cells, they provide strength and do all the basic function. So in short, basically, there are 200 different types of cells, and each of them is completely different. So although they are completely different, is their genetics totally different? The answer is no, because all of them started from the single cell. And if you look at the genes, if you do the sequencing of the DNA, you will get exactly the same DNA pattern in any of these cells that you pick up. So what causes this immense diversity? If the genes have, if the only answer were genes, then basically we would have been just a lump of 40 trillion cells, but that's not the case. Every cell is different. And also, not all the cells are in same numbers. There are certain cells that are very rare in the body. Then there are certain cells like skin. It's in billions and trillions. So what's the difference? what causes this immense diversity? It's definitely not just the genetics. So epigenetics is a very broad term which comprises of many different phenomena in the body. So anything, basically anything that happens on top of genetics. So here you have this, there are the same genes in all the cells, but they are different. So that phenomena is basically epigenetics. Now, I will briefly talk about how the DNA, how the chromosome, many people often are confused between the terms like what is DNA, what is chromosome, what is chromatid. So I will talk briefly about these things. And for that, we'll start with a cell. So in this figure, you can take any cell, and the basic structure, basic architecture of any cell is similar, if not the same. So you have a cell body, and inside it is the nucleus. So nucleus is where all the instruction, where all the DNA is present. So inside the nucleus, you have DNA, and that DNA produces proteins. So protein is a very generic term. For a common man, protein means what we get from food. But protein is much more than that. Any component of our body. For example, you have skin, or you have a bone, or for example, anything, the hormones that you have, insulin is also a protein. So for a human body, the protein, for human body or for any life form, the word protein is very diverse. It refers to any compound that is composed of 20 of the different amino acids. So how are those compounds produced? So those compounds have the blueprint in the DNA, and the DNA resides in the nucleus. So there is a blueprint, and from that blueprint, every time the body requires a protein, it produces it. So inside the human cell, inside the human nucleus, there are 23. In this talk, just clarify it in the beginning. So in this talk, all the data and all the numbers and everything that I will talk of is mostly related to humans. So when I'm saying 23 pairs of chromosomes or any other things, it's mostly about the humans. All the other mammals have the basic architecture and the basic the fundamentals is pretty much the same, but please take it as human when I'm talking. And please feel free to stop if you have any questions, or if you don't understand, or at any time you feel that I'm going fast, or there is anything which I couldn't clarify, I'll be happy to stop and answer, because the whole point is to understand, to get a flavor of what the field is, rather than me just giving a presentation and some formal question and answer at the end. So please feel free. Yeah, so so each chromosome is basically a long chain of 4 nucleotides. So what is a nucleotide? It's just a chemical, which it's just a chemical compound, and I won't go much into it. So it's basically every chromosome is a long chain of 4 it can be of 4 nucleotides. They are adenine, thymine, cytosine, and guanine, and they can be in any order. They can be AATTCC or ATAC, AG, that can be in any permutation and combination. So what is it? That is a chromosome. So basically the entire length of a DNA, that's a chromosome. So what is a genome? So in humans, as you know, there are 23 pairs of chromosomes. So again, coming back to this stage. So when the life starts with single cell, you have one chromosome that one set of chromosome that you inherit from your father, that comes from sperm, and one set of chromosome that you inherit from your mother. So we have 23 pairs of chromosomes. You have inherited one set from your father, another 23 from your mother. So inside the cell, you have 46 chromosomes and 23 pairs, because 2 chromosomes, basically 2 similar type of chromosomes are alike, that we call homologous anyway. So every cell in the body. So when and also when you look at like the 23 chromosomes, you might have often heard, like in human beings have 3 billion base pairs or 3 billion nucleotides. So the 23 chromosomes have 3 billion nucleotides, which we call the sum total of this chromosome and everything is called genome. Now, every cell in the body has exactly the same sequence of nucleotides. So if you take, say, there are 23 pair of chromosomes, if you say, and those are named as chromosome 1, 2, 3, up to 22, and then you have sex chromosomes, which is XX in females and XY in males. So for example, if you take chromosome number 1, which you have inherited from your father, you take you sequence it from your brain cell and you sequence it from your kidney cell, they would be exactly the same. Oh, sorry. They would be exactly the same. Then what makes cell types so distinct? So here is the answer. This is how, it's not an answer, but this is how the chromosomes, the 23 pair of chromosomes, they are inside a nucleus. So this is the nucleus, and now we are focusing in a nucleus. So the red part that you can see, it's a very simple schematic. So that part that you see, it's a stretch of DNA or a stretch of chromosome, which is basically active. So from this part of the chromosomes, the proteins can be made. While the part that is strongly condensed and they're in black, from these parts, the proteins cannot be produced. So the difference lies is, so when you look at a kidney cell or a liver cell or a neuron or a heart cell, so the regions in a DNA that are open or basically accessible to make proteins, they are different. So for example, there is, say, I don't know if there is an option of pointer here. I don't know. Does anyone know if there is a pointer option here?
I think you can use the cursor to mark anything.
Okay, I'll do
The
that.
marker, yes.
Okay. Say, how does it work? Okay, so say we take one part of this which I have drawn here. You can see in green. Let me change the color. Yeah. Okay, so suppose we look at this part in a nucleus. So this is the part of the gene, this is the part of the DNA which is open and is accessible for making the proteins that are required. So suppose this the area that I have drawn is are certain number of genes on chromosome 1. That gene might be available for making proteins in a heart cell, but in the brain cells, that particular part might be condensed and some other part might be open. So that's what differentiates different types of cells. Then there are certain part of the genes, there are certain proteins that every cell requires. For example, proteins that the cell requires for basic survival, basic metabolic activities like glucose processing or making functional like basic survival proteins which you require for division of the cells. So those parts will be open in all the cells. But then there are certain very specific part, like the part for like how do neurons have electrical conductivity and skin cells don't. So the proteins that are responsible for that, those part of the gene, those part of the chromatin might be are open in nerve cells, but not open in other type of cells. So that's what differentiates the two different kind of cells. And this is a phenomena which epigenetics, as I said, is a very wide phenomena, and I will come to each of those phenomena, each of the major phenomena, because talking about all of them is beyond the scope of like 1.5 hours of talk, basic talk. So I will give a flavor of most of them. So yeah, this is the basic part why there is a difference. And so here we start. So has anyone a question? Okay. So this is how so the chromosome that we picture. So basically it's if there are 23 long stretches, 23 pairs of long stretches.
Do we have any questions for the speaker now?
Could we mute? There's some background noise. I don't know where it is coming from. Yeah. Okay. So this is how I talked about that every human cell has 23 pairs of chromosome. So now I will start what exactly is a chromosome. As I said, it's basically formed of different like 4 types of nucleotides: adenine, thymine, cytosine, and guanine, which is ATCG. And this is how they are arranged inside a nucleus. So each of the A, that is adenine, that binds with thymine, and each of the guanine, which is G, binds with cytosine. And this is the long backbone. It keeps on replicating. So you have a long backbone, which is basically the double helical structure. This is a simple straight line, how they are basically attached to each other. And if you look at the structure of this part, so this is the helix that you always see. So this is not always in a straight, this is not in a straight line. So they form a helix, and this is how they are arranged inside a nucleus. So every chromosome that you see is basically complete. It starts with this, and it's just a repeating. It's a single stretch of this double helix. So this is the double helix, and you have basically 46 or 23 pairs of these completely long double helix. So every time a cell divides, so you have the similar thing. So it goes into two similar, it makes into two similar. So you have 46, that 46 becomes 92, and then they segregate into 2 different types of cells. So again, those 2 cells, the same process keeps on repeating. That's how you have a parent DNA. And then in the cells that you have similar DNA. So this one of the strands, so this is double stranded, because I'll again, it would be good if I annotate it. So yeah. So what we are looking here is one strand, and so this is one strand, and this is the complementary strand. So every time there is a replication, so one of the strand acts as a template. So here, as you can see, there's one strand which is acting as a template, and then there's another strand which is exactly like this strand. So for example, if this is acting as a template strand, then when there's a division happening, replication happening, so the complementary strand will form exactly like this. Similarly, when you treat this as a strand, then its complementary will be like this. So every time there is a cell division, all the 46 pairs of DNA, all the 46 DNA, the 23 pairs, they are exactly the same. They are inherited exactly the same in each of the cells. So this is the basic about the DNA. Now I also mentioned briefly about protein synthesis. Like every cell needs proteins for all its functions. So how do they synthesize protein? So you have DNA. So from DNA, there's of course replication, and then you have transcription. So the transcription is a process when you have this template of the DNA. It's the genetic blueprint. So from that genetic blueprint, this RNA is formed. And RNA is the primary template from which the RNA is basically the secondary template, or the direct template from which protein is synthesized. So the question would have been, why is not RNA? Why do we not have RNA as genetics? So it's a complicated thing, because RNA is not stable, and also DNA is much more stable. You already have a blueprint. And whenever you need to whenever the cells need to make proteins, they first make an RNA, and from RNA they make a protein, because it's a very regulated process. But if you know, since these days, people might have read a lot about Corona and COVID. So the COVID virus, it has RNA as its genetic material. And RNA is the same thing also in the RNA vaccine, you might know. So we are injected an RNA, and that RNA basically makes the protein that we need to fight off the COVID virus. So that's the antibody that is generated after an RNA virus is injected. So I think if you have any question regarding this, or if you have not understood, please feel free to ask. Otherwise, I'll move to the next slide, which is, oops, why is it? How do I delete this? Okay. Yeah. So Again, as I said, this is how the DNA looks. You have this DNA, which is a double stranded. It folds into double helix. But you know, there are 3 billion, 3 billion and in 23, so that means 6 billion. So even if the space inside the nucleus is very small, it is in the terms of few microns, which is like 10 to the power minus 6 meter. And if this is that long, like you have so many billion base pairs, they cannot stay in like completely long elongated form. So how they are managed, how they are arranged in the nucleus. So they are very tightly compressed. And how are they compressed? So they are compressed with, again, these are proteins. So this is the form, like on the right side of the slide, you can see beads on a string. This is the DNA. The first, just look at the right side of the slide. So the first is how a short region of a DNA double helix looks. So now if you look at this entire region, it is complex. It is basically a double helix, and it conforms like this. Now each of this is again basically tightly coiled around a bead. So what is a bead? These beads are also protein, which I will come later. Then how does it further fold? So these beads and everything further folds among itself. Like for example, if you see these 3 beads, they are again super coiled into this structure. And this is how the super coil is further. If you look at this, this is totally coiled here. And then this is coiled. This is very small part, as you can see. So this is how tight as a long structure like DNA. So if you can see, only this part is 2 nanometer. But if it was completely long, it would have not fit in the cell, in the nucleus. So they are tightly coiled, tightly compressed. And then this is how they are inside the nucleus. But then the problem arises. If they are so tightly coiled and they are not accessible to protein manufacturing machinery, what happens? How do cells produce the machinery? How do cells produce protein? So the answer to that is that whenever the cell needs to produce a protein, a certain part of the chromosome, so this is basically chromatin. So this part of the chromatin gets opened up. So that part is not very tightly packed. As you can see here, for example, in this case, the parts of the chromatin that are not tightly packed, that determine which type of cell they will make. Coming to the previous part, I talked about there are beads, and these beads are different types of proteins. So what type of proteins are these? We call these beads as histone. So there are 4 types of histone proteins, basically 5, but inside the core, there are 4 types of histone proteins, and you have 2 copies of each histone in each of the cores, so making it 8 histone proteins. And this is not a very static structure. These are very dynamic. So when the cell needs to produce a protein, these tight structures, they can be uncoiled. So this is called uncoiling of the DNA. So they can be uncoiled, or when the cell doesn't need it, they can further make it tighten. So there can be coiling and uncoiling of the tight structures of the DNA, which continues throughout our life cycle, because this is a very dynamic structure. And this is moderated through various different types of enzymes. So what they do, you can add certain chemicals, you can add certain methyl groups, you can add acetyl groups. So these, when you add these groups, people who have chemistry background might be aware that these are small groups like the methyl, acetyl, sumoyl. So these are different groups, and you can add these groups covalently, or you can remove these groups covalently. So when you remove or add these groups, that changes the conformation of the DNA. So a part of the DNA where these groups have been added, they can either be open or they can be closed. Depending on that, the same thing that I was talking in my previous slide. So here I will present a schematic. So this is the DNA. So as if you look at this, so going back to this slide. So say this is a DNA, this is a stretch of DNA. So this stretch of DNA, say if this particular DNA has, say, chromatin 1. So if the chromatin 1 has 100 million of nucleotides, so that might be divided into certain, say 1000 genes might be there on that particular chromatin. So the stretch of, so if this is a long DNA, DNA is basically the same as chromatin. The long stretch of DNA is one chromatin. So when someone says that there are 23 pairs of chromatin in our human body, that means basically we have 23 pairs of long DNA structure in our body. And on each of those chromatin or on each of those DNA, we have many genes that reside on that. So a stretch of a DNA is basically a gene. So say this is a chromatin and there are 3 genes on it. So during the developmental time scale, what happens is, say in a muscle cell, the gene 1 is switched off. Gene 1 does not express at all. It gets tightened and there is no expression of protein from it. While genes 2 and 3, they make proteins, which are very specific to, say, muscle cells. Now, similarly, if you look at what happens if the gene 1 is shut off, gene 1 is open, gene 2 is on, but gene 3 is shut down, then you have different set of proteins in the same cell. So again, remember that the DNA is exactly the same in both the muscle cell and the skin cell, but they are different because one of the gene is shut down in muscle cells while the same gene is open, and on the other hand, say the third gene is shut down in the skin cells. So this is how different cells are regulated. And this is not a very like a fixed process happening only in different types of cells. This process also happens in a similar type of cells. For example, when there is cancer happening, when someone has a cancer in liver or lungs. So what happens is, despite all the cells being a liver cell, some of the liver cells have epigenetically been misregulated. So ideally, the genes that should not have been turned on, they have been turned on, or the genes that should have been ideally switched on, they have been switched off. That leads to an erratic cell division leading to cancer. And all these processes are called, they fall under the broad category of epigenetics. Now I will get basically the term epigenetics, and the next few slides, since I think I have given a basic flavor of how DNA is, how genes are, how proteins are translated, how the basic in a cell happens. So now I will take you through epigenetics. The word epi is it means on top of, and genetics refers to the study of genes and their functions. So if you say someone is genetically predisposed to a certain disease, that means that the two people might have completely different genes. For example, when we say that Europeans are genetically different to Asians, or even among Asia, we have different genetic groups. Or even if you zoom in, so the genes between your different family members are slightly different. But when there are changes which are happening above the gene, so even when the genes are similar, like the examples that I have been giving inside our human body, inside a single human body, that is called epigenetics. So genes are the blueprint that we inherit, and epigenetic is the changes or the modifications that happen on top of these genes. So when the talk started, Pratyushda mentioned a very interesting term that is very, very commonly used, that nature versus nurture. So genes is basically the nature. Genes is what you inherit. And epigenetic changes, the modifications that happen on top of those genes, that is the nurture part. So what are the epigenetic mechanisms? Development, as I was talking for the last few slides. So in your childhood, in the uterus, so when one cell divides and differentiates into multiple different types of cells, that is an epigenetic. When you are exposed to certain chemicals, you are exposed to certain drugs, pharmaceuticals, you are exposed to pollution, smoking, stress, or any of the lifestyle changes, that also changes the epigenetic landscape through different mechanisms, which I will also discuss in the next few slides. Aging is another thing. Aging is a very strong influencer of epigenetics. So there are many studies, and I'll also present a slide where if you look at the chromatin of a young 3-year-old kid and compare it with a 100-year-old or a 50-year-old kid, the same gene, the same chromatin is completely different. Certain parts of genes are switched on, certain parts of the genes are switched off. Now, what are the mechanisms? So in this slide, I'm just talking about one of the mechanisms, as there is a methyl group. So methyl group is a carbon with 3 hydrogen atoms. So that methyl group may be attached to the DNA. So there's a DNA, and most of these methyl groups attached on cytosine. So you have 4 amino acids, A, T, C, and G. So these methylation, whenever happens, so the methyl group gets attached to the cytosine. And this is a very interesting phenomena. So an attachment of a methyl group on a DNA can completely switch it on or switch it off. The other changes can be the changes on histone. So there are 2 sets of changes. One can be addition of groups just on the DNA. The second can be the addition of groups on the histone proteins that basically wrap around the DNA and give it a tight coiled conformation or uncoiled conformation. So the health endpoints, basically these can lead to cancer, these can lead to autoimmune diseases, several mental disorders, diabetes, metabolic disorders, and a host of other disorders, which I will just talk about in the later slides. So this is what roughly epigenetics is. The entire changes that you see on top of the gene. It can be on the DNA, it can be on the histone proteins. So what these happen, these regulate the proteins that can be switched, that can be produced in a cell. So as I said, the epigenetic modifications can be either on the DNA itself. The most common of it is the methylation of the DNA. Or, as I said, there are 8 core proteins that attach, that basically form the core of a nucleosome. So the bead on the rings, the structure that I showed. So this bead is basically made of 8 proteins that I said. They are histone. And those histones are named H2A. H stands for histone. So you have H2A, H2B, H3, and H4. You have 2 copies of each of them, making it 8. So any of these histones, like H2A, H2B, H3, and H4, they can be modified. So how can they be modified? So you have these host of modifications that can happen. So let me annotate it a bit for you. So this is DNA methylation. So what happens is this part is the DNA. So there is a methyl group added to it. The second one that happens, which is very common, is histone methylation 3. So what is 3K27? It's a very simple thing. Just please pay attention and I will explain that to you. So H3 here refers to the third histone, the H3. As you can see, here is H3. So this is a modification on H3 histone. So K refers to an amino acid called lysine. So there must be an amino acid lysine on the 27th position of this protein. So what happens is 3 methyl groups are added to that particular lysine on histone 3. So this is one modification. Similarly, again on histone 3, at the 9th position of lysine, there can be 2 methyl groups. This can be there, this cannot be there. So these are the modifications that keep on happening throughout the life, depending right from the day the single cell is conceived to your death. Basically, these changes keep on happening depending on the needs of the body. So I'm not getting into the locations and everything because it gets a bit technical, but what I can explain is the function. So when it is silencing, so silencing means when this modification, so when 3 methyl groups are added on the 27th position of lysine in third histone, H3, that particular gene basically gets silenced. So for example, if this entire thing is a gene and there is H3, there is a 3 methyl group added. So what will happen is this mark will tell the protein manufacturing machinery or the RNA making machinery that, see, I have put this mark. Now you don't have to make RNA or proteins from this. And that's how it will be shut down. So the genes has not changed. You have exactly the same gene, but now despite having the same gene, your body does not produce the protein that the gene accounts for. Similarly, these are different types. This one has focused more on the H3 methylation because H3 is a very interesting methylation. H3 is a very interesting histone where a number of changes take place. Similarly, this is, I'll talk about this one, and this is an extremely interesting, the one in red. This is a very interesting epigenetic change. So what happens, it's again the same thing, same lysine. Here, if you can compare these two, H3, same lysine, same position. Now, instead of 3 methyl groups, if there is an acetyl group, what happens is it leads to uncoiling of the DNA because DNA is negatively charged and so is the acetyl group. So when due to the charges, so these are basically attached on lysine. So what happens is the structure of the DNA completely changes, and from the tight coiling, it now completely opens up. So these two are very important epigenetic changes that might lead to either silencing or activating of a gene. So, okay, so these are the epigenetic changes that happen, and these are just a few.
Can I interrupt you for a moment?
Yes, Pratyush.
Can you repeat again what are the factors that decide between epigenetic changes, please?
Okay, so you mean the environmental factors,
yes,
stress?
yes.
Okay.
Yeah.
Okay. So most of the changes that are beyond our control are the changes that happen during our development. So
Right.
that's beyond our control. Then the next part is the nurture part, basically, which is environmental exposure to different food, different chemicals, different pollutants. So there was a study, I mean, since you asked this, there was a study, the plastic that is used to make most of our bottles. So they contain a compound called Bisphenol A.
Right.
So that Bisphenol A is one of the strong epigenetic modifiers. So what Bisphenol A does is it methylates a number of certain developmental genes, due to which there is a increased risk of cancer. So this was found like a few years back, 3 or 4 years back, or 5 years back. And then what European Union did is that it banned the use of Bisphenol A in production of any plastic, because in adults, Bisphenol A is not such a big problem, because even if we take it, the amount is so low that the evidence that it might be doing some changes is very low. I mean, there might be some evidence that is building, but in infants, it's very strong. So if the infants are getting exposed to even lower doses of Bisphenol A, it exposes them to a risk of cancer, because this Bisphenol A changes their it adds methyl groups on certain parts of the genes, and that leads to cancer. I mean, it increases, not every child who drinks from the bottle that has Bisphenol A will develop cancer, but it increases the risk. So European Union banned the use of Bisphenol A in all infant bottles or any of the packaging products.
Right.
That's
So
one change.
does it make sense to think that our genetics are more mutable when we are closer to birth? Because this is something that you said very early, that
Yes.
a mouse which is very close to we were talking before the talk started, and you said that 3 days, still 3 days, they can recover. And when you say this, that the adults are less susceptible to these changes than infants, does this give a general idea that we are more flexible with respect to exhibiting our epigenetics when we are younger compared to when we get older? Is this a general?
Yes, it's exactly the case, and there's a very interesting study. Those who are history buffs here might be knowing about the 1944 famine in Netherlands, when the Nazi Germany basically blockaded everything, and Netherlands did not have access to food, and there was a huge famine. So we also talk about that study where it showed that children born during that period had a lot of problems, and there were epigenetic reasons behind it. Which is your guess, or basically your understanding, is quite correct. So these changes affect much more Because the reason is in humans, the development keeps on taking place up to a certain age. And when you have developed, say, after basically the adolescent part, most of the genes, most of the neuronal connections, most of the other things have stabilized. And with metabolism, during the growing phase, everything keeps on changing very quickly. So that's quite true.
Okay, that's very interesting. Okay, sorry, continue.
So
Yes.
I'll talk about that as well. Yeah, the next.
I think we have a hand raise from Srikant, so he might
Where can I see the hand raises? I should be able to track.
in the
these.
participants list, if you can see against the name of the person.
Right, I see. Srikantoda, yes, please.
Yeah. I mean, coming to your question, I mean, what Kshitij was explaining, I'm being a pediatrician. I'm a little bit intrigued. Probably what you were telling in reference to bisphenol, probably the relative amount of toxic material in infants will be more as compared to the adults. That is one factor.
Yes.
And the second factor, even in younger infants, they're amenable to changes since they're not stabilized, isn't it?
Yeah.
Yeah, but
that's true. Because in infants, the dose of bisphenol that is inherited. Also, the reason is that infants are mostly bottle fed, and they keep on, due to their small size and due to their more exposure to the bottles, the bisphenol they have in their system is much higher than what an adult would have. In addition, so it's a double whammy basically.
Thanks.
Yeah.
Thanks.
I have one query.
Yeah.
So cancer, is it more of a genetic related disease or more due to epigenetic changes that happens after birth?
If this would be convincingly answered, I think we would get the cure. Cancer is the most heterogeneous disease. So just
is this correct? Kshitij, is this correct that the problem of cancer is it's actually not a disease? It is. Then we have to describe what disease means. But from what I understand, once your cells go berserk and they do not know when to stop and they do not have control anymore, that's cancer basically.
That's true. Yeah.
Yes.
And I have done like a lot of genetic studies where I've looked into cancer cells. So the problem with cancer cells or the cancer as a disease is that even from the same person, from the same tissue, for example, if there is a patient, or even if there is a mouse model where you have a biopsy, very small size of biopsy of a, say, liver cancer, that the problem is that even a single, we call it basically nodes. So even if you take one node, it's genetically so diverse that you cannot say that this cancer has been caused due to exactly the same factor. Some cancers are a bit less heterogeneous, like a breast cancer, where you can pin down on certain mutations like BRCA1, and there are some other genes. There are some certain type of lung cancers where it's still easier to pinpoint that there are certain genetic mutations or changes in the epigenetics. But in general, cancer is very difficult to pinpoint on any of the cause or any of the genetic factors or any of the epigenetic factors. It's a hell lot of complications to understand what cancer is. That's the challenge that every molecular biologist, every geneticist has, and also all the clinical practitioners as well, because when they treat a cancer with certain drugs, every drug basically treats a certain type of or certain mutation or certain type of mechanism which is leading to cancer. But the point is that the cells are so heterogeneous, you treat it with 2-3 drugs, and then there is one subset of cancer tissue which is kind of either resistant to that drug or simply is not, that drug simply doesn't have any role in treating it, and then it keeps on multiplying. So it's a very difficult, cancer is a very difficult to study as well as to treat, definitely, because if you know the exact cause of something, most likely it's easier to treat or sometimes easier to prevent. But with the cancer, its biggest problem is its heterogeneity, in my opinion.
Some tobacco companies, they keep on arguing that governments have been very critical of them without having any scientific basis of attributing cancer to tobacco directly, because again, people who don't consume tobacco, they also have mouth cancers and all. Tobacco being an epigenetic external factor that happens after birth, and again, there are people having mouth cancers without having any exposure to tobacco. So how relevant is their argument?
It's not really, again, there are 2 points here which we are kind of missing. First point is regarding, I'll first answer your question on the tobacco companies. The point is, it's not always that. That's a very simple or very gullible, very simple way to mislead the population, saying that, see, those who don't consume tobacco, those who don't smoke, they also develop cancer. So the very simple answer to it is to look at the incident rates. There are a lot of studies that have been done, and tobacco companies have been they have been either asked to put that was the reason why they were asked to put in big photos like that causes cancer. Anyway, that's a political thing, and I'm not getting into politics and industry political nexus. So the point here is science always works on evidence. And how does that evidence work? The evidence works on large cohorts. So there are studies of, say, tens of thousands or at times hundreds of thousands people who smoke versus those who don't smoke. And then you also control for confounders. So confounders is basically, you compare 1,000 smokers who are very rich and have good access to medical health care, have good food, to 1,000 non-smokers who are very poor, or who don't have, who work in, say, asbestos factory or silica factory. So in that case, even though they don't smoke, their workplace exposure to silica or other dust might be making them predisposed to cancer. So when a scientific study is done, you control for all these factors, and then you basically look at whether the incident rates. In short, basically, if you are looking at 1,000 people, 1,000 patients who smoke and are from a rich family, have good access to medical, have similar genetic makeup, then you will choose exactly 1,000 people from a similar socio-economic background who are not smoking. And then if you look at the incident rates, there is much difference. Like those who smoke, they are absolutely, overwhelming evidence. There is no one who can even say that smoking is not associated with, cancer is not associated with smoking. So these are the things we must always keep in mind.
100% sure?
Excuse me?
Are you 100%? Sorry, but yes, only for a fun interruption. I'm a smoker, so I would like to hear something.
Yeah, it's, there's a huge correlation is very huge. As a physicist or a mathematician, correlation would be a good word I should use here. So between smoking and the point is, many times people say that, see, in West, people smoking, in Western countries, almost everyone smokes, and still their average lifespan is longer. But in India, people. So the reason is that, again, when you compare average lifespan in India of people who are smoking and average lifespan of people who are smoking in the West, the problem is you are not accounting, you are not balancing for the socio-economic factors.
Right.
And the second point is, of course, even if you are a non-smoker in Delhi or Lucknow or any of the Indian cities, you are exposed to pollution. You are exposed to so much pollution that, these studies are always very difficult. I'm not saying that these are black and white. These studies are very difficult. There is so much of confounder effect that it's difficult to regulate and control for all the effects. But yes, when there are controlled studies from like similar cohorts and similar socioeconomic conditions, similar geographical locations, then the evidence is overwhelming. Yeah.
Right. Continue, please. Yeah.
Okay. So these were the epigenetic changes that I briefly discussed. And now I will start with some of the interesting phenomena apart from development, apart from diseases, which I'll come also later. So first is X inactivation, X chromosome inactivation that happens in females. So what exactly is X inactivation, X chromosome inactivation, or the X inactivation? if you look here, this is, everyone knows here, I think that's not, I have to go into detail, that a female, every cell in a female has 2 X chromosomes, and every cell in a male has 1 X and 1 Y chromosome. The interesting thing about X chromosome is that it's huge compared to a Y chromosome. It's really huge. I mean, if you look at the size, I mean, you cannot see much difference on this picture, but just to give you an idea, I mean, sorry, it's a I wrote it wrong here. There are about 910 genes on an X chromosome, while there are just 50 genes on a Y chromosome. So you can understand how it's close to 20 times, 50 and 910, like 19 times, 18 times bigger. So the X chromosome is really huge in all aspects. And these genes, the genes that are present on X chromosomes, they are not just genes that lead to sex determination, because X is something that is common in both males and females. So X has body functions. X chromosome has the genes, those genes make proteins that are required for basic body functions, including many of the growth and day-to-day normal activities. Now, if in a female, both the X chromosomes were active, the females would be producing 2 times, every cell in female would be producing 2 times the amount of proteins than in a male. And that would lead to basically completely haywire system. So how exactly does it happen? So what the body does is, in every female, one of the X chromosome is shut down completely. So there is no protein formation, there is no transcription. Transcription means making of RNA from the DNA. None of that phenomena is happening in every X chromosome. So you have 2 X chromosomes, one is switched on, one is completely switched off. And this happens in all the 40 trillion, I mean, not all the 40 trillion cells, because the red blood cells don't have, basically it doesn't have a nucleus, but anyway, they do have genes. So basically in all the cells, one of the X chromosomes is switched on and one X chromosome is switched off in female. Now, how does this happen? So when basically a female is born, when there are 2 X chromosomes inherited and the cell division starts, the development starts, the baby starts to fall, our body randomly starts switching off one of these X chromosomes in every cell. And that's how it happens. So how it happens, I'm not going into much of the molecular aspects. So there is this is a structure of a chromatin. Say, for example, if this is the X chromatin, there is certain protein that regulates that one of these should be completely switched off. So that protein binds to it, and then it starts to modify the histone. Histone is basically the beads that I talked in the previous few slides. And once it modifies, it recruits another protein, and the role of that protein, it's an enzyme basically. That protein or that enzyme, what it does, it starts to compress the chromatin, and it keeps on compressing it. So once there is a chain that starts, and that wave of chromatin condensation. So condensation is just compression. So that goes on throughout the X chromosome. The only difference is in X chromosome, one of the marks that is here is not just a protein, but sometimes a single-stranded DNA or RNA could also be an epigenetic regulator. So what happens is there can be a complementary sequence of an RNA, or which we call transcript, that can bind to this particular DNA. And then the body realized that, okay, that means since there is a complementary binding, I need to shut it down. So then what it does, it starts to recruit other regulatory proteins that might simply start this wave of chromatin condensation. So this is how X chromatin is shut down. This is one of the most important epigenetic factors that happens and that regulates our body, because there are certain diseases or not diseases, disorders, genetic disorders where X chromosome fails to shut down. For example, there are certain trisomies, there are XXXY, where there are where a women, or I don't know if you call them women or whatever, they just inherit three X chromosomes and only one is shut down. So they have 2 X chromosomes where different genes are active, and they end up with many disorders, and there are many syndromes, Klinefelter syndromes and others. I don't exactly remember which one is which, but there are a few host of other syndromes which are associated with Downs, Klinefelters. Yeah. So I don't recall the names exactly, but yeah, any doctor here would be well versed with this. Now, the most interesting thing which Pratyush said in the start of the talk, like, are the two identical twins? Why are they not exactly like 100% similar, even if they have exactly 100% same genetic makeup? So here also, it's just like inactivation of X chromosome was an epigenetic factor. Diseases are regulated by epigenetics. Development is regulated by epigenetics. Same happens here as well. So here, I don't know if you are, these are very famous Bachelor twins. They do some singing and dance, and they are very popular on YouTube. That's why I chose the photo. Most people would be aware of them. They are monozygotic twins, basically, which means that there are two twins. In common languages, they are called either identical twins or non-identical twins. So identical twins are basically when there is a single cell, and that single cell has been fertilized, and then it develops into two babies. On the other hand, non-identical twins are, there are two eggs, there are two sperms, so they are just like two babies, and they are twins just because they are born at the same time. But apart from that, they are genetically dissimilar. Dissimilar in the sense that they are as genetically similar as you are with your sibling who might be older or younger to you. But the identical twins are exactly the same, because in most cases, what happens is one cell goes on to form one human being. But in this case, something happens that one cell forms two babies, and they keep on developing. So basically, they both come from the single cell, and they have like 100% same genetic makeup. So ideally, if they have completely the same genetic makeup, they should have exactly the same incidence of diseases. Like, for example, you are in early 70s and 80s, there used to be a very popular statement, you are what your genes are. And that led to all this human genome sequencing and everything. But that view is changed now. So if you are what your genes are, then in that case, if one of these children develop autism, then others should definitely have developed autism. Or same, if one identical twins develop diabetes or cancer or schizophrenia, the others too should have. But that is not the case. So if you look at the concordance rate, so concordance rate is percentage of twins having a same disease as the other one. So if you look at diabetes, the concordance rate, although it is high, because of course genetics is also one of the factors. What my point is, genetics is not the only factor. So here the concordance rate for diabetes is 50%, while it's, I think, less than 5% or less than 2% for normal twins. So of course, due to same genes, there is an increased rate of concordance, increased concordance rate for diabetes or schizophrenia, but it's not overwhelmingly same, like it's not 100%. So what exactly is the difference? Why is there so much difference despite 100% of the same genes? Not just that, also they have difference in many other aspects, like they don't always, not all the twins grow up to be of same size or the same, they are not socially similar. They have many different things. Their predisposition to other social factors might be completely different from each other. So these things are in short called nurture. So this nurture, everything that is happening at nurture, it goes to affect your body at some level or the other, because nurture is, even say someone has a different behavior, so that behavior is not a very abstract thing. So the change in behavior basically registers as a change in the neural connection that you have in the brain. Someone might be more aggressive, someone might be less aggressive, someone might be more emotional. So if someone is more emotional, that is not because of something just abstract. They are more emotional because the part of the brain that accounts for emotions, which is amygdala, might be that part of the brain in one of the twins is slightly more active than the other twin. So why does this happen? Because after they are born, despite the fact that they are at the same, they are being given access to everything similar, not everything is exactly the same. Maybe due to the random processes of epigenetics, which, as I said, that during development, there are certain epigenetic changes that are beyond our control. That is just because the body does those changes. Then there are certain changes that might be due to environment, hormones, stress, or the way you treat them, or maybe due to different kind of food. So with time, due to multiple of those factors, they start they are epigenetically reprogrammed despite having the same same gene. So for example, here, if you look in this slide, they start with the same gene, but the affected twin might have some epigenetic changes, and due to that epigenetic changes, he or she might develop some disease or some other. It might not be disease; it might just be some phenotype, which is just, as I said, about behavior. It's not a disease, but someone might be. So that there's a strong epigenetic correlation to it. And was it just an hypothesis? No. So there have been now a number of studies on twins where scientists have looked at their epigenetic marks, where they have looked at the chromatin condensation. And if you can see here, so there's both panels of images are from, they were published in 2005. It was published in Proceedings of National Academy of Sciences. It's a very reputed journal in our field. So this there they looked at the chromosomes of different age, and as you can see, even at the age of 3 years, when you look at the chromosome number 3, the twins have totally different chromatin makeup. So if you look at the first panel, at the lower end of this chromosome, it's much more yellow, and at this end, which is green. So these are just false color. So one color can represent the part of the genes that are more active, and the other color might represent the part of the genes that are shut down. So if you look at this, for example, here, this part of the gene on chromatin 3, although this is from a twin, this part is, if yellow represents more active or it's more shut down. So here it's more shut down, more part of the gene is shut down, but that's not the case here. Similarly, there's a difference on the different regions of the chromatin. Again, if you look at the 50-year-old twins, there is a difference, and this difference is much higher. So if you look at the 3-year-old twins, there are certain genes that are certain part of the chromatin that is in the process of transition. So green is active, yellow is they are basically transitioning to switch on or switch off state, and red is a complete switch off. So as you can see, there is not a uniform switch off even in the 50-year-old twins. There has been a different, so which means that this is all epigenetics. Ideally, their chromosomes, if you sequence them, they will be exactly similar. But at the epigenetic level, at the chromatin level, there is no similarity at all.
So Kshitij, one question: why is the left picture so blurred compared to the right?
Because these are very, chromatin is on like a nanometer scale, and if you add false color with staining, this is the best technological images that you had at that time.
No, but why does it get so better towards the right when the guy turns 50 and not?
It's not better. It's just the color. It's just the color code that you have. It's the image is exactly similar. There's a big blob of red due to which it looks like more.
Yes.
Yeah. It's.
I see. But so there are better indications when you turn older.
Yes, I mean that is. I'm coming to the next slide in that as well. So that's a more recent study where they looked at twins who are 3 years old, or not twins. That's normally between a normal cohort of 3-year-old people and 50-year-old compared it with 100-year-old. I'll show that as well.
I see, but it makes sense. Okay, so things are better segregated as you get older. Right. So
aging. That's what. So I'm coming to the 4 important factors which. I have missed one of these, but anyway, 5th one is the age. So 5 factors that influence your epigenetics. First is random epigenetic changes, as I said. So as you are developing and throughout the life, there are certain epigenetic changes that are, you can call them stochastic because you don't have any control. Even if you keep two twins or 2 people exactly in the same, you give them 100% identical environment, they still will be different because in human body, there will be certain changes due to that's just pure randomness, which will make the epigenetics different in two people.
Right. Can I
Diet,
interrupt you one more time, please?
yeah.
I see a question from Rakesh Govil that I did not see before.
Oh, sure.
Maybe you have answered it already, but can you explain the terms writers versus erasers? This
Okay,
has
sure.
been a long time.
Sure,
back.
I'll do.
My
I'll do
apologies.
that. I'll do that. I'll go to the previous slide, and it will be easier for me to explain it. I think he is talking in one of
Mr. Rakesh, can you take over and just
This-
ask a question yourself, please? Rakesh? Rakesh Da, Rakesh Jr.
I think he has
I
left.
think he is left. Oh, my apologies. I did not pay attention.
No
Sorry.
worries.
So either you can answer for it. This is a good question. I think you could just
Okay.
answer for us. Yes.
So I'll explain. I'll answer it. So writer is basically any mark. It's not a very technical term. It's a very simple term to, for example, if you are adding a methyl group on a DNA. So what you are doing is you are writing methyl on top of DNA. So enzymes that do this thing is called a writer enzyme.
Right, I see. Okay.
And if you are adding, say, histone, if you are adding an acetyl group on a histone, that's again you are writing on it. So that's a writer group. And then similarly, there are erasers. So you erase the methyl group that was already on the DNA. So what you do is you remove the methyl group. So for example, in case of methyl, or for example, acetyl, if you are adding acetyl, writer would be a histone acetylase. This is the name of an enzyme, which is a writer enzyme. So what it does is adds an acetyl group. It writes an acetyl group. So histone acetylase is a writer enzyme.
Right.
Deacetylase, it's another enzyme which basically deacetyl. It removes the acetyl group from the histone. So that would basically be an eraser enzyme or an eraser protein.
Right.
Then you have proteins that just maintain it. For example, if there is a methyl group on a DNA or acetyl group on a histone, if you don't maintain it, it might be either washed off, or sometimes you just need to maintain it for a certain number of time. You need to protect it. So then there are also enzymes or proteins that you just call maintenance protein. So writer, eraser, maintainer, these are very simple terms. These are not scientific terms. So you have scientific enzymes, which can be, in case of you can have DNA methyltransferase, you can have demethylase. So I mean, these are different types of enzymes, but this is a very generic term: writer, maintainer, and eraser proteins.
Right.
I hope it clears.
Yeah.
Okay.
To me, it does. Yeah.
Okay. So again, random, and then the second is diet. We don't know how the food we are eating. I mean, we don't know the mechanism, but we definitely know that the food we eat is one of the strongest influences on our epigenetics. The reason being that although we don't know exactly how the food we eat changes the epigenetics, but we definitely know in certain diseased conditions like high blood glucose patients that they have different histone modifications. This is something that our lab does, exactly like the lab I work in. They are also working with. We are working in a cardiovascular division, so we also get human blood samples. And so once we get human blood samples, we look into many of these immune cells that are in the blood, and look and compare it in the case of diabetic patients versus in case of patients that are very healthy and don't have any diabetes or metabolic disorders. And we see that there is a change in histone architecture, the chromosome architecture in these patients who have high blood glucose. So definitely there is an association. Epigenetics is a very recent field. I mean, in field of molecular biology, anything that is 20, 30 years old is very recent, because you take like 5 years, 10 years just to study a single phenomena. And then it's such a vast thing. There is so many food, there are so many epigenetic modifications, there are technological challenges that we are still on our way. We don't know exactly the mechanisms. We don't know convincingly how, excuse me, how these changes occur at the molecular levels, and if we can explain it, we can pin down to the exact molecular causes. But yeah, there are strong associations that we see, and we hypothesize that these associations are caused by the food changes, and further studies are needed to confirm the, as I mean, in science, you know, correlation and association is not a convincing causation. You cannot just say that. You can always say that we see association and we hypothesize that these changes are related, but to convincingly say that yes, this causes this, we need to pin down to the mechanisms. And same is true with the chemical exposure. We are continuously exposed to pollutants and chemicals, gases, and these also lead to changes in methylation, acetylation, and complete remodeling of histone complexes. And again, these are also very complex fields, because the point that whether it's a cause and effect, like whether if you are smoking or if you are getting exposed to it, whether it's exactly the pollutant that is causing these epigenetic changes, or these are secondary factors that changes because of there is some other influences of smoking or pollution. It's very difficult to say the direct, because I mean, there are a lot of studies still going on. These are studies in progress. I mean, people might say that epigenetics, the term was coined in 1940s. That's true. But at that time, the meaning of epigenetics was completely different. The modern concept of epigenetics as a molecular biology that I'm talking about right now and what we are studying is pretty recent. I think it was mid-90s or late 90s. I'm not very good with dates here, but it's about 20 to 25 year old, or maybe 30 year old at the max phenomena. And the fourth is, of course, social interaction. Everything, every social interaction, everything that you have, you are happy, you are stressed, you undergo a trauma, whether it's physical or mental, your social life, every factor affects your epigenetics, most likely. And for example, increased stress or trauma during pregnancy have been conclusively linked with children which have long-lasting adverse phenotypes. They might have metabolic disorders. They themselves might be stressed. They might have some other diseases in their long life. And again, these are these you find from cohort studies. You take women, for example, you take 100 patients or 100 women, pregnant women who faced domestic violence, or they had some financial stress, and then take other 100 women who had better access to everything. They did not have scarcity of other thing. They were not harassed. They were not sexually assaulted or any such thing. And then you do follow-up study with the children as they develop into the teens, and you see that there is a link between these two studies. And as again I said, this is still a developing field, so we will still have that's something which is a problem with human patients. So when you are doing a human patient or a cohort study, unlike mice, where you set up a breeding pair, a mouse is born in 18, 19 days, and then you follow it up for 2 months and 3 months, and you have a result in 1 year. In human, it's simply not possible, because you cannot set a breeding pair in human and say, okay, I have 100 breeding pair and I can do whatever I want. I mean, you just have to wait for it, and then you have to do all the control trial, you have to do the double blinding, and human trials take like, they go into decades. And then after decades, you just come to know about the association, and then you do the further follow-up studies at the molecular level, which is yet another level of complexity, and it takes ages to convincingly get detailed answers for these. But there was a convincing study, and that was the study which basically fueled all this growth. And that is one of the most interesting study that I'll be talking and I'll be telling you all about at the end, because I should have started at the beginning, but I thought I should preserve the most convincing part of epigenetics for the end of the talk. There was a Dutch hunger famine in 1944 when the Nazis blockaded Netherlands, and it could not import food grains from different parts. Its economy had already been ravaged after the bombing and other stuff. So it was basically left to itself to import food from other places, and importing was blockaded by navies from other side. So at that time, there were mothers who were malnourished, and these malnourished women gave birth to offspring who were perfectly healthy at birth. That's very interesting. I mean, these mothers had, there was a famine. They had very limited access to food, and despite that, they gave birth to a perfectly healthy. The good thing with these people was that the famine at that stage was not like the entire year. So these women had access to food at certain later part of the pregnancy, like the last 4 months, 5 months, and that's why the children were born pretty healthy. The only difference was that these children were slightly smaller and weighed lesser than what an average Dutch children weighed in that era. Now, a large proportion of these children who were born smaller, interesting thing was that when they became older, like in their 50s, 40s or 50s or 60s, a large number of them developed obesity and a number of other metabolic disorders. In contrast, the incidence of obesity and other metabolic disorders in children who were born to Dutch mother who did not face food scarcity during the famine was significantly less. I mean, isn't it interesting that what the children faced when they were not even like grown, when they were just in their mother's womb, that part affected their growth many decades later in their life. And these children, those who were born with the mothers who were malnourished. So these children, they also reported greater incidence of several other health issues such as mental health and other metabolic disorders. So what happened here exactly? So what exactly here happened was that I will just try to understand. I'll be going a bit slow, and I have not written, I've not made any diagram here. So, for example, when a child is growing in the womb, and there is very less access to nutrition because the mothers had scarcity. They could not get the complete food that they wanted or that they should ideally have gotten. Then due to the limited food that they got, there was an effect on their insulin-derived growth factor 2, which is a very important growth factor, which regulates our growth, which regulates our size, and regulates a host of our body processes, including metabolism. So in that environment, when they had less access to nutrition in the womb, the body started to recognize what body did. That body started to recognize that, okay, I am getting very less nutrition, so I should make the best use of it. So the metabolism changed at the epigenetic level. That epigenetic marking that the children had in their womb, that epigenetic mark stayed throughout their life. So what happened, even when they grew old, they did not have any scarcity of food, and they had like lot of food. So now what body realized, that the body had already those marks, which was making the best use of nutrients, even with a limited amount of nutrient. Now, when they got a lot of nutrient, the body started making use of those nutrients as well, which I'm explaining it in a very crude way, not going into the mechanical and molecular details. So what happened, that they started to grow obese because their body were primed or programmed in such a way that they could make the best use of nutrients, even if the nutrient was limited. So for example, if you get 100 grams of food and you are able to metabolize all the 100 grams very efficiently. So that is a good thing to happen when you have a food scarcity. But for example, someone who gets 100 grams of food, but their efficiency is only say 60%, like they can only metabolize 60% of their food. So in that case, if they are grown as an adult, they have their metabolism is a bit slower because they only metabolize 60% of their food. But in these cases who had scarcity of food, their rate of metabolism was a bit higher, and that metabolism rate was epigenetically imprinted throughout their life. So as an adult, when they did not have access to food, did not have any scarcity, they completely, like, they kept on putting on their weight because the body could metabolize everything. That was an epigenetic mark on insulin-derived growth factor 2 gene. That was one of the genes, and the scientists looked at other genes as well, and there was a host of other genes that had been epigenetically affected in these children who had whose mothers were exposed to famine due to the Dutch hunger famine in 1944. This was one of the most interesting things, which basically opened this field and showed that epigenetics is, it continues basically what happens at the time of your birth, or not even before birth, when you are in the womb, it goes on. That's why, I mean, if you have heard people, doctors ask women not to take stress or to drink less, because it not only affects you as a mother or as a parent, but it also affects the children, not only in the short term. I mean, of course, there are certain things that affect the complete developmental growth of children. There are certain chemicals that directly go into the bloodstream of children and stunt their growth. But then there are certain changes that are happening at an epigenetic level, at a very slow level. And those changes manifest much later in the life, when the child basically becomes an adult. So this is what I was talking about. So for example, the epigenetics part are not just like random. They are it is sometimes inherited, as I said, like these children in this case. So there were some epigenetic changes that happened when the children were in womb, and these epigenetic changes were inherited. So how does it inherit? For example, generally in a DNA you don't have methyl group, but say one of the cytosines was methylated here. So what happens when there is a replication going on? This methyl part is recognized, and when it is recognized by again, there are readers and writers and erasers. So these are proteins or the enzymes which reads it when replicating the DNA. It reads that, okay, in front, say adjacent or complementary to my strand, there is a cytosine which already has a methyl group. That protein reads it. So when it synthesizes the new strand, that reading mark is already there, and then that reader recruits a writer and asks it to write it in front. So here you have this, like this is on the extreme left, you have the original methylated DNA, and after replication, this methyl group is added here. Because when you replicate a DNA, methyl is not added. Methyl is a modification that happens after the DNA has been synthesized, after the DNA replication has been taken place. So now there is one more interesting phenomenon. As I said, like there can be some random stochastic modification in the DNA. So what is that random thing? So for example, this was the original DNA on the extreme left. I will annotate it here. So suppose this is the original DNA inside a cell. Now there are two methyl groups here. The body, this is the template, and here you have the template. Now when the DNA synthesis is happening, because DNA is always complementary, so you have this strand, and then there will be a complementary strand in the other cell. So when the E is synthesized here, complementary to it, there will be no methyl group. So what will happen when the DNA has been synthesized? The DNA will recruit a reader. So that reader will read that there is a methyl group here. So since there is a methyl group here, there should have been one here, because these are complementary marks. Now this reader will recruit a writer, and that writer will add a methyl group here. That's something you can see. And now you can see this one and this one, they are now completely identical. Now, sometimes what happens, just randomly, you can call it a failure of the replicating machinery. Either the reader is not recruited, it's just a mistake. Although these mistakes are like one in many, many trillions, but if there is a mistake, sometimes these mistakes can be harmless. Sometimes these mistakes can be very deadly. So But these mistakes are very rare anyway at the first place. But sometimes these are the mistakes. There can be mistakes at many places. There can be mistakes even at, for example, you have A and T, but instead of T, the body can add the body can add some other thing here. So that is a genetic mistake. That is a mistake which is at the level of gene. So when there's a replication, there can be a mistake at this, but this is also, there are many checkpoints. So out of 40 trillion cells that are replicating constantly, and they are replicating and you don't get many mistakes. I mean, and once in a while you get a mistake and that might lead to say cancer or some other thing. So similarly, same thing happens with epigenetics as well. Just randomly there can be an epigenetic change because your machinery forgot to read or write it. Even sometimes if it read it, it forgot to write. Similarly, same thing happens here. If you take this as a template, there is here, and this has to be added, and this is added. And again, you get this and this, which are exactly the same. So there is one epigenetic mark in one cell, but when this cell divides into two, you have these epigenetic marks, which have been faithfully inherited by the two cells. And this is how exactly it happens.
So can I come down and get it to my room? All right, great, thanks. All right, okay, thank you.
Okay. So now there is a very interesting, I think I have talked about three or four phenomena. The fifth interesting phenomenon with epigenetics is imprinting. So imprinting. In human bodies, as I the starting, like there are genes, set of genes you get from your father, one set of genes that you get from your mother. So there are certain genes which are only active when inherited from father, and then there are certain genes that are active which are inherited from the mother. So those genes are not active from the paternal chromosome, but maternal.
If I don't spill all of it, bring something at least.
Okay,
Are you that drunk?
I'm drunk.
Really badly drunk, is it?
I'm drunk.
Okay, so
Okay, this is your test. You handle things in life. You can walk from here to the
Yeah,
door.
I can.
Okay, beautiful. That's all.
okay, so some genes you have, like they are inherited directly from dad, and some genes directly from mom. So if both of those genes are active or the wrong one is active, that leads to certain diseases. So how does this happen? So body basically has some epigenetic marks on this, and it knows which gene to activate. Should I activate that gene from the father's chromosome, or should I activate that gene from the mother's chromosome? And this is very faithfully maintained throughout the life. This is not for all the genes. Again, I'll make it very clear so that you're not confused. Most of the genes, like human body has about 20, every cell has about 25,000 genes. So all of those genes are active from both your father paternal chromosome as well as your maternal chromosomes. There are less than or close to about 100 genes which are only specific either from the paternal genes or from the maternal genes. So these genes are epigenetically silenced on the other chromosome. So for example, if a gene has to come from the maternal part, it is silenced on the paternal part, and vice versa. This is also an interesting epigenetic phenomena that regulates our well-being. Now, when epigenetic imprinting goes wrong, this is very similar to, this is a study in mice. Again, as we said, we cannot manipulate humans. It's of course beyond ethics, and of course no one would be willing to get it done, these studies in humans. So we have to make mouse model. So in mouse, what we do, like there is, again, we have come back to the same insulin derived growth factor, which is a very important growth factor. So in mouse, what happens is, so this growth factor has a receptor. So this is the growth factor, and this growth factor has an IGF-2 receptor to which it binds and then starts the signaling so that it regulates the growth. So the gene for the IGF-2 hormone, that is from the mother's gene, from the father's gene, and the gene and the receptor to which it binds, that is from the father's gene. So in mice, there have been studies. So what you do, you just mess it up. So you either change it, you manipulate the system, you either make both of them switch on, or you make the wrong one switch on. And then what happens? So if you delete the mother's one, you have a huge offspring. If you delete the father's one, you have a very dwarf offspring. And because this gene is very specific to which chromosome it comes from, because this one is imprinted. Similarly, there are diseases in humans that are caused by abnormal imprinting, which are like, I'm not going into it, Beckwith-Wiedemann syndrome, Prader-Willi syndrome, and Engelmann syndrome. So these two, the second and third Prader-Willi and Engelmann syndrome, these are due to the changes, I think, not exactly sure. It's either chromatin 14 or chromatin 15. Most likely it's 15. So there's a region of that chromatin which has to be, which is either some part of it is from father, some part is from mother. So if there is a mess up with that system, the system is messed up and it leads to these syndromes. Now, the fourth aspect is age. With age, there is a change in methylation level. So methylation on the DNA. I don't know if you can see it or not. This is a plot basically where they have done the sequencing to determine the methylation percentage. The inner circle is I think it's a newborn. The outer one is 25-year-old, and the most outer, the external one, is of an adult or an old man who is 101 years old. Now you look at the percentage of methylation in our DNA. Here it's quite high in the newborn, and it progressively goes down as you age. And in the 101-year-old, the methylation has been completely down. As you can see in the graphs as well, they have looked at chromosome 2, they have looked at the chromosome 8, they have done the proportion of methylation in the CpG methylation. So here you have like 82% of the CpG island is methylated in. So CpG island is just like, I'll show you, it's not a very complicated term. So everywhere in our human body, the C and G basically comes together. So this repeat of CG is called a CpG island, where you have this CG rich region in the genome that is called, and since the methylation is always at the cytosine, so you calculate the methylation at the CG regions. So this is what they have calculated. They have looked at the CG region and what proportion of those CG island is methylated. So this is, and there's a very significant decrease. So
one more question again. There is
yes.
no methylation at A and T at all,
No,
is it?
no, not at
all. Okay, thanks. Okay.
So methylation is always at the cytosine.
Right, okay, thanks.
It's a very simple, I mean, I didn't get into the chemistry. So if you look at the structure of all these purines and pyrimidines, it's only the cytosine which can be, I mean, you can do the reactions. You can, in a test tube, you can make, you can add cytosine, you can add methyl group to any of these. But in human bodies, due to the structure of the DNA, the other arrangements are not practically possible. That's all. I mean,
I see. Okay, great. Thanks.
so in human body or any of the mammalian cells, it's the methylation is mostly at the cytosine.
And cytos means C and G together.
No, cytosine is C.
Okay, G is guanine.
Guanine, yeah.
Right. Okay.
And these, cytosine and guanine. So you have these adenine, thymine, cytosine, and guanine.
Right.
And these are further divided into purines and pyrimidines, and there. So I'm not getting into chemistry because it's not relevant to the talk. Okay, so
okay, great. Proceed, please.
Yes. Yeah. So here you can see they have looked at the chromosome 2, parts of the chromosome 2 and parts of chromosome 8, and there's a difference in methylation here as well, which shows that, which is very distinct. It's not that there's any like dichotomy, or it's very clear that the methylation is completely going down. And they also looked at the distance to the closest CpG site, and here also there's a strong correlation. The red one is with the old man, and the blue is for the newborn, and this distance correlation also changes. So this was the basic phenomena about the epigenetic, what it was. And now I will briefly, very briefly, just in one slide, since we were discussing and at that time not many were there. So I will talk about what I am doing currently here at in Oxford. I will start with the basic thing that in mice, first I'll just give a brief overview of heart attack, myocardial infarction, what exactly happens. So in human heart, whenever there is a heart attack, what we call as heart attack is that there is a blockage in one of the arteries. So when there is a blockage in one of the arteries of heart, this leads to the death of cells. So the cells here that help in beating the heart, they die, due to which the function of heart function is reduced. And sometimes when it's very severe, people die because the heart cannot pump blood anymore to the body. So this is what a heart attack is. So what happens in heart attack is, when there is a heart attack and the blood flow is disrupted, the cells there start to die. And when the cells there start to die, that part is replaced by a scar tissue. So that scar tissue is basically fiber, which we call technically as fibrosis. But this is what happens in all the adults, in mammals and human beings, and all the adults, all the mammals. But interestingly, what was observed a few years ago was when you look at a young mouse pup. So you take a young litter in mouse and they are like just one day after they are born. So when they have a heart attack, interestingly, even when the cells here die after heart attack, all the healthy heart beating cells, they regenerate themselves. It's very interesting because this phenomena stays only like 3 to 4 days after they are born. And after that, again, it's same thing happens in mice as well. When you induce heart attack in these mice, they have scar tissue there, and the healthy beating heart cells, which we call technically as cardiomyocytes, they do not regenerate. So what interesting phenomena happens between like first few days and after like 7th day, that in the first few days, all the heart can healthily regenerate, but that phenomena, that capability to regenerate is lost after 7 days. So my focus is to study the epigenetic aspect of this. What epigenetic changes happen between the first 7 days that leads to these first 7 days that helps us distinguish the differences between the mice heart at the 1 day post birth and 7 day post birth. So to study that, what I have done is I have created a mouse model. So what that mouse model is, I will P1, every P means postnatal. So P means how many day after birth they are. So P1 means 1 day old mouse, P7 means 7 day old mouse, and PI is post injury. So I will tell briefly about the experimental plan, what I do here. I take mouse, and the day they are born, I do some surgery. So what is that surgery? I do that surgery to tie the heart in the mouse. I tie an artery. So once you tie an artery, it mimics heart attack. So that artery is tied and the blood cannot flow anymore to that part of the heart. And there, basically, you have induced a heart attack. I do the same thing of mice when they are 1 day old and when they are 7 day old. And then I follow them 1 day post injury and follow them 7 day post injury. Then I isolate the heart. I kill the mice. I isolate the hearts from them. I isolate spleen from them. So why spleen? Interestingly, because many of the regeneration that we see in heart, that is mediated through immune tissue, immune cells. So we have macrophages, we have neutrophils. These are all different kind of immune cells. So whenever there is an injury, whether it is an external injury or whether it is an internal injury, you have immune cells that are mobilized. And where are those immune cells mobilized from? These immune cells are mobilized from bone marrow. These immune cells are mobilized from spleen. And of course, they are already circulating in the blood. So I'm looking at these organs. I also harvest these organs from all these 6 different age group of mice. So we have one mice which is without injury. So this is a control. So we see whether the changes that we are happening post 1 day post injury, whether those are genuine changes. So there must be something to compare. So in science, the control is very important, and the control here is a baseline, a mice without any injury. Similarly, we take a control for a P7, which is a baseline. And then the next plan, once we have done it, once we have isolated it, we prepare single cell suspension, which has a suspension with all the cells inside it. and then we do the sequencing. And sequencing is a very, I'm not getting into the technical aspect. It's just that we look into the epigenetic landscape, how the epigenetics has changed between first few days and in different conditions as well. This is basically the technical thing that I always do. So I do the dissociation. Basically, I mince the heart and dissociate into single cells, and then isolate the nuclei directly. And then there's a process called library preparation, where we generate the libraries that have to be sequenced. Then we sequence it. There are sequencing machines. Then we analyze the data. And once we analyze the data for this sequencing, which we call assay for transposase accessible chromatin. So in short, it's a technical term. In short, it just looks at the open and closed part of the DNA, the closed part which cannot be accessible for protein manufacturing machinery, and the open part which can be accessed. And then we investigate the chromatin changes in the landscape of chromatin between the first day and the 7th day. And we also investigate the changes that are happening in the same animal, but across the different cell subpopulations. So for example, you have different types of immune cells. So how is a macrophage different from a neutrophil in the same mouse in the same age group? So that is the second level of questions that we are addressing here in my lab. So that is the basic that I talked. I don't want to get into the detail because if I want to talk, it would be too much detailed about the sequencing and technicalities, and it would be beyond, I think, most of the people here who are not very technically familiar with this sequencing and other aspects. So the last part of my talk, like I have just 2 more slides, which is these are the things that are happening in our body. But everything that we study has to have some translational role. Like even if we know something, there is an epigenetic aspect behind it. So how can we help it for human being? How can we do our best to understand that knowledge that we have gained from epigenetics and put it to mankind's benefit? So for that, what the ultimate aim of epigenetic studies is to try to reprogram the epigenetics, try to reprogram the cell, reprogram the chromatin, reprogram the genes that have been shut down. First thing is, it's very difficult to change your genes. I mean, there's a totally different thing with gene therapy that is happening at the genetic level, but this is even further than that, which is epigenetic reprogramming. Can you epigenetically reprogram certain part? Can you rewrite the methyl on the DNA? Can you rewrite the acetyl groups on histones? Can you rewrite the sumoyl groups? Can you rewrite the methyl groups on histone? Like there's so many modifications. Can you rewrite on different kinds of histones? Can you modify it using some drugs? Can you modify it using any external input? So that's the ultimate aim that field of epigenetic wants to do, like change the epigenetics at different level. I mean, it's a very it's a very futuristic thing to be talking about, because we don't even know about epigenetics in most of the cases. We don't even know the mechanical changes that happen in the epigenetics. So talking about the drugs and talking about externally modulating these epigenetics is a very long-term thing that science is planning on. But anyway, so have we got any breakthroughs in it? Yes, we have. Today, there are like a bunch of drugs which target epigenetics and they target the reprogramming. So there are 3 broad categories. So what are these? Histone deacetylase inhibitors. I will first explain the name without getting into the name of the drugs, because name of the drugs is immaterial here. So histone deacetylase. What is a deacetylase? Deacetylase is something that removes acetyl group from the histone. So someone asked me, what is an eraser? So histone deacetylase is an eraser. So what you do when you inhibit an eraser? You stop erasing it. So if I have an eraser and I want to erase something and you take the eraser away from me, I cannot erase it now. So this is what the first class of drugs is trying to do. It is trying to block the eraser itself. So once the eraser is blocked, it cannot erase the epigenetic marks. And these are like 5, 6 FDA approved drugs, which are not just like on trial, but some of these are even administered to people, and most of them are in leukemia, which is blood cancer, colorectal cancer, and prostate cancer. And if you can see, most of these are in these three types of cancer as well, except I think there is one, yeah, there is a panobinostat, which is also used for head and neck cancer, and breast cancer, and thyroid cancer. Similarly, the second type of drugs that are in use is DNA methyltransferase inhibitor. So this one is a modifier of, this stops the modification of histone, but this stops the modification of methyl on the DNA. So DNA methyltransferase is something that adds methyl group to the DNA. So when you inhibit a writer, so methyltransferase is a writer. So when you inhibit a writer, you cannot add a new group on the DNA. So these are the four drugs that basically do the same thing. And finally, there is just one drug which is targeting histone methyltransferases. So it basically is again it is inhibiting a writer. So that writer adds methyl group on a histone protein. So these are the drugs which are showing good results. They have been FDA approved. But again, as somebody said, cancer, somebody asked about cancer, and I answered that cancer is so heterogeneous. There are gene therapy going on. There are epigenetic drugs. There are so many other things going on. So it's difficult to, the point is different doctors, based on their success rate, different protocols, they do a mixture of these drugs depending on genetic studies, depending on sequencing, depending on different results that come from sequencing studies of cancer samples. And one last thing, it's about this slide only, but how do they do? As I said, when you basically inhibit a writer, they cannot add the group. And what does acetylation do? So when acetylation is basically opens the gene, but now when the acetylation is reduced, it does not open the gene and it closes the gene. So this part, how you do, you target it with the tumor suppressor genes. So in our body, there are many genes. Why do we not get cancer in normal day, and it's very rare to get cancer? Because there are many genes which suppress cancer. So it regulates how many how cell division has to happen. So those genes are called tumor suppressor genes because they control cell division. They do not let your cells multiply erratically. So what you do in cancer, these genes are kind of switched off. So the tumor suppressor genes have been suppressed. So you reactivate those genes by basically reducing the acetylation, and that in that particular part of the gene where it is happening. Similarly, there are pro-cancer genes. What you do, you shut down the expression of those genes which are causing cancers. So how do you do that? You do that by writing more methyl groups. The more methyl groups you write, the more compressed, the more condensed the DNA in that part becomes. And then you do a mixture of these two things. You suppress the oncogenes, you suppress the pro-cancer gene, you suppress the pro-cancer proteins and activate the tumor suppressor genes. And this is by a mixture of these two things. This is a very basic thing, and you can see the class of drugs that are of those two, and they generally use a mixture. And I'm not a doctor, I'm not a trained medical practitioner, so I am not saying anything medical. This is just scientific. This is how science works at a very molecular level. And I'm not even, I do not have any knowledge about the dosage and which drugs are to be given in anything. So these are beyond my competency. These are beyond my training. So please do not take these slides as medical advice. And it's a disclaimer which I have to put because this will be in public domain and people might think it as a medical advice. And every time we present it in a public domain or anywhere we use these things, we have to make it very clear that this is just a scientific phenomena that we are addressing, and this is not a medical advice, or this is not a medical consultant, any such thing related to medical. So hope you understand that. And if you have any such question on medicine, please consult a doctor. Yeah, thanks a lot. And if you have more questions, I'm open to discussing or answering anything.
So thank you, Kshitij. This was a wonderful talk. Beautiful.
Thanks, Pratyush.
I had many questions, which partially I understood. I will ask you some more questions, but before
Sure.
I go, the floor is open to the audience. Questions, please. The youngest guy first, who is the host? Mr. Satyam Kumar. Did you hear the talk well?
Yeah, sir. I think it was very elaborate, almost a biology class that we missed
Yes.
after a school. I am first time actually listened for a long time for biology because I took science of engineering after 11, 12. So in, I have like one question again related to cancer. So epigenetic changes that have happened over the years. So as you, one of the things that you mentioned that in older people, the epigenetic changes are relatively lower, or am I
No.
correct? No.
No. What I said that the methylation levels are lower, which means that more changes have happened. So if you look at the methylation levels that are much higher in a newborn. So methylation is the mark. So there is a mark on a DNA.
Mm-hmm.
Methyl group has been added to the DNA. So that percentage, that level of methyl group is higher in the younger children. And that level has gone down. So basically more epigenetic changes have happened. If there was no epigenetic change, then you would have expected the methylation level to remain consistent throughout the age.
Okay.
So can I ask you a very dumb question?
No question is dumb, Pratyush.
So, as we see that I understand hydrocarbons. So CH3 is the methyl group that we are talking about, right?
Yes.
This is sort of a, I would say it like once a methyl group comes, nothing else happening. If CH4 is like what I see, carbon and carbon has like these things. So the basic group is the methyl CH4 is the smallest carbon.
CH4 is methane. So you
methane, right? Exactly. Yes.
So carbon has you have carbon, and carbon has 4
Yes, I understand this. So methane was the name I was looking for. Let me stop you now.
Yes.
So methane removing one hydrogen becomes the methyl group.
Yes.
And methyl group, when it attaches to something, I think from what I understand very dumbly, it's not allowing any other thing to happen. Is this some sort of a protector of genes?
It's not, I mean, it's
because
not.
What I understand from your talk, the lesser methylation means this is directly proportional to our aging. The more methylation you have, the younger you are, apparently. And we also know that with age, there is something called the telomere. You will understand this better than me. So this
I didn't get into that. Yeah, I mean, it's a very, it would have it would have opened another can of.
I am opening this for you, right? Because this gets me curious.
Okay, yeah.
Apparently, so for the non-initiated ones, you can tell me if I'm wrong or not. So telomeres are the end sequences, the end parts of the gene. And when they are somehow not able to maintain their sanctity is when the genes, they start reducing in size. And this is directly proportional to our aging. And is it correct in that case that methyl groups somehow are responsible for shielding this breaking of genes into small, you can phrase it better than what I'm doing right now.
Okay, I think I got your question. I think
Yes.
question is, I'll explain to others as well, but before answering you very specifically. So your question is, are the telomeres protected by the methyl group, or is the
not only telomeres, the whole of genes, whole of genes. Yes, telomeres maybe specifically also, yes.
No, in short, answer is no. So methyl group is not acting as a protector. So why telomeres start to get shorter is, I'll tell you, it's a complicated thing, because it's something which you can understand if you try to draw, if you look at the process of replication, like you have a DNA, you have 2 strands of DNA, and when it replicates, there is a primer attached to it. That primer, and then it builds on basically a new. So this is a template. You have A, T, G, C here. Then there is first a small primer attached here. From that primer, you will have a build-up.
Right. Right.
And that primer, what happens every time the cell divides, that primer is basically cut off. And so the shortening happens due to the number of replications. So every time there is a replication, there is some
I see.
DNA that gets shortened because it was used as a primer. So that is not because of methyl group. So that shortening is happening at a different level. That shortening is happening because you have, when you look at the replication, so in replication, that part of the gene is basically cut down. So with every cycle, and at the end, what happens is, so at the end, the telomeres have completely gone, because every protective part has now been got after, like, say, if a cell goes around 40 rounds of replication, that 40 round of replication, the telomere has been completely gone now. So that cell basically will cease to exist. And that's why telomere has a very important role in aging and senescence. So once all the telomeres has disappeared, basically, the DNA can no longer replicate like truly, because now if it starts to replicate without the protective end of telomere, then you will start losing the genes, because main part of the DNA. And there's one more thing which people are not very aware, that like we have 3 billion nucleotides if you look at the 23 chromosomes, but and there are about how many genes? There are, human body has about 20,000 genes. Gene has maximum, I would say, 4,000 to 5,000 nucleotides. So basically it adds up to a few million. So why are there 3 billion nucleotides? So basically only 2% of our gene is what, 2% of our chromosome or our genome is what codes for genes. The rest 98%, earlier there was a term that was used for it. Earlier we used to call it junk DNA. But now
I strongly suspect it is not junk, but
it's not junk. It's not junk. So that's a very recent, like last 8-10 years, this field has opened up. So what that part of DNA does is that, although it does not regulate, it does not manufacture proteins in itself, it regulates the nearby regions which are producing.
Yes. And the reason why I ask, okay, no, continue. Sorry, yes. And because we are going to hijack this talk between you and me unless others have questions.
Yeah, I mean,
but continue, please. Yes.
Yeah. So the part is, so the point here is that if the junk DNA, it used to be called the junk DNA, but now what we call is, there's a term for it. We call it long non-coding RNAs, or non-coding DNA fragments.
Yes.
Because initially it was thought that, okay, they are sitting in the system without any use. They don't make any transcripts. They don't make any protein. They are just remnants of evolution, where we started as a single cell and then it just kept on getting bigger and it kept on adding DNA, but it did not have any function.
But I don't think this is the correct story. Yes.
Yes. So that's not the correct story.
Again, you will correct me. Let me interrupt you. I started looking with somebody about how genes replicate, and I only do it through the mathematical perspective about where is the site of replication. You will understand these terms. I do not understand clearly.
That's called origin of replication.
Yes, the origin sites of replication. And not only this, apparently what I learned from this guy, there is a whole mechanism. When a gene has to replicate, first it unfolds. Yes. After it unfolds, there is a machine that comes and sits at the first place. Behind this machine, there is a host of other machines. This guy is going tuck, 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 tuck. If it makes a mistake, there is another machine behind it tells it you made a mistake in replicate. Transcription is the word. When there is transcription.
No, I mean, it's not transcription exactly.
Go on then.
So transcription is when from DNA you have RNA.
Right. So DNA replication. Replication is
replication. Yes.
Sorry. Yes. So right.
That's called a replication fork, where you part. So as I showed you, DNA is totally coiled. First it's helix.
Yes.
The first helix is coiled into proteins like histones. Then that histone part is further coiled.
Yes.
Again, you have like a complexity at completely different level. And then what happens? You unfold it at completely different level. And after unfolding it, there are proteins called helicase. So
Right.
helicase DNA is a double helical structure.
Right.
So it opens the helicases.
Yes.
And then there are, and it's such an interesting, such a wonderful machinery that it
Yes. Yes.
randomly open it at anywhere. It
Yes.
opens it very close to the origin of replication.
Yes.
And then the machinery sits there. And once it has sat there, the machinery, it starts adding, and if you have, since you read it, you might be knowing that the addition of a new group is always on the 5' side.
Yes.
So
Yes. 5, 3, 3, 1, and
Yes.
5, 1 is what we are talking about. Yes.
yes. So it's just like if you look at the sugar and carbon, it's just
Right.
the
Right. Yeah.
1, 2, 3,
Yeah.
4, 5.
Yeah.
So it's always at the 5' side that you add a new group.
Right. Yes. Yes.
And this is a very interesting phenomena. This is a very interesting phenomena because you add and interestingly, you have a long DNA. If there are many people who, and I don't even blame, or I don't even blame them or say them they don't, because they think that it opens at 1 and it just keeps on
No.
getting
No.
to
No.
that.
No.
So
There
what
is a
happens is
host-
it opens at
host of
a host
machines
of
that-
different places.
This I can even tell. I started we are looking at this mathematically, and apparently, so to the uninitiated, I will give the mathematical side to this. If we think of a very big novel, let's say Isaac Asimov's Foundation, or Lord of the Rings by J.R.R. Tolkien, these are 50,000 words, maybe 100,000 words. And each word, if we think, let's say on an average, maybe 10 letters long. We are talking about 1 million letters here. If we wanted to copy this blindly, we will make mistakes. Already in these 10 millions a lot. But genetic replication, it's maybe 10 out of a 3 billion. That is the mistake that we are talking about, or 20 or maybe more. You can tell me the number.
Not even that. Not even that.
Not even 10 out of 3 billion. Not
Yeah,
even
not
10
even
out of
that.
3.
No.
So to
It's-
the uninitiated, you should tell them what I'm talking about here. You will be able to explain better.
I don't even have the numbers, because the reason is that there are so many cells. We have like trillions of cells. Many of them are replicating at the same time.
But it is almost exact copy, and
Yes.
3 billion means it is a whole library that we are talking about.
So-
Not even one book.
I will tell you, 3 billion just, have you seen like in a normal novel that you have?
Thanks for explaining. Go ahead. Yes.
So if you take a normal novel, like a normal demi-size novel in which you have written, so if you just write 3 billion of those with the same spacing that they have, then all the 3 billion in our genome in one cell, that is just in one cell.
Yes.
So that length will basically exceed to, I think, close to 2,000 kilometers. From Kolkata to Delhi, it's like
Yes.
1,500. So that's the length if you just write at a normal length. So that's, and when you replicate, and that is just in one cell, and the body has trillions of such cells. And when it replicates it, it hardly makes a mistake.
It hardly makes a mistake is the important point here. This is extraordinary.
And when it makes a mistake, I mean, they do make mistakes at times.
Something is behind them telling them you made a mistake. Come back and correct.
And that is, and if so, there are many types of mistakes that happen. Although the number is very less, but many types of mistakes they do happen. So it can be at the replication, like for example, if there is A is always attached to T, G is always attached to C. So instead of A, in place of A attaching to T, the body attaches A to say C, which is untenable. It would lead to whole lot of diseases. And so what the body does here is that it reads it, it realizes that it has made a mistake. It goes back there. There is a separate machinery for it. It removes C from there, completely digests it, adds a T to it, and then again a link, like kind of as a sellotape, it completely what do I say? It's a very beautiful phenomena. It cuts it, it takes it away, adds C there, realizing that it made a mistake, and then again ligates it. So it's a enzyme is called a ligase because it's basically ligating it. It's like a loose end of, because when we are replicating, DNA is like one chromosome is like millions of bases are long. Now, even if you have made one mistake and you have cut it, so one of the end becomes loose because it's no longer a double helix like a ladder. Imagine a ladder here. If there is a ladder and you have put something wrong, you cut one side of the ladder, then basically the ladder is now open because it's no longer ladder attached to the. So it adds it and ligates it completely. And this happens at all the places. Now, where certain places where the body realizes that it cannot replicate it. So then what it does, it starts another set of signaling events, and then it recruits a number of other cells. Basically, it recruits a number of immune cells, and it directs it, say, I have messed up with this cell, just
eat it.
Don't let it stay in the cell.
Beautiful.
Every day, like in human body, I think I read somewhere that few thousand cells are being eaten every day by our immune cells, protecting us from cancer. Some day in the life, if one of those cells escape, therefore, I don't know if you have, I think you might be aware of it. There's a immunotherapy. There's a new branch which people are trying to apply in cancer. So the role is to train the immune cells towards, just like you train a sniffer dog to drugs and harmful. The aim is to train the immune cells to recognize all kind of immune cells, all kind of cancer cells, and then inject those immune cells. Take your immune cells, train them to different kind of cancer cells, and inject them in the body if they can recognize the cancer cells.
This is extraordinary. Yes.
I mean, cancer is so different, and that's why people are trying so many different approaches, because with virus and with bacteria, the good thing is that you can simply, you know how it works, and you can just do an antibiotic. If one doesn't work, you can try with another, because you know the mechanism, what exactly is going on. But with cancer, as I said in the starting as well, it's so heterogeneous, you don't even know what is going on. So you try to address it at multiple levels. You try to address it at immune level, you try to address it at genetics, you try to address it at epigenetics, you try to address it at just cell division level, or at the protein level, you try to block those proteins which are causing cell division. It's so, if you don't know the answer, you're trying to address it at multiple levels.
Right. Another question, maybe you know this about it. Is there a correlation between cancer and body size of animals? I have heard that, I cannot again ask you a proper question, but apparently the idea was that the more number of cells you have, the more possibility of some of the cells going rogue. And so larger animals should have more instances of cancer, but this is not what they see.
No, it's not true. In fact, elephant is the only animal which doesn't.
Yes, and this is extraordinary, because in general you would expect that, because if you have more cells going rogue will give you a larger chance of developing cancer. So you expect that larger body-sized creatures should somehow be more prone towards cancer, but this is not how it goes.
That's not true. That's also that's another field of study, and people are studying, like they're taking cells from elephants and trying to figure out not just elephants, other animals as well, which basically, and there's I may be wrong, because I cannot say this very convincingly, but I don't think there is a correlation between body size and
no, there is not. No, this is what there is not, and this is actually surprising in a way,
because there are also smaller animals. I think there is a type of I think there's a spiny mouse or something which can regenerate, and it doesn't have incidence of cancer. I'm not sure which one is it, but there are some small animals as well which don't have cancer. There are large animals like elephants as well which don't have cancer.
Elephants have no cancer at all.
Very, very rare, like almost, you can say it's almost none.
That's very interesting.
Hi, Kshitij.
Hi.
Hi, it is Anand there. Ananddeep. Yeah.
Hello, Ananddeep.
Yeah. How are you? Good to listen to you. Yeah, just a quick thing. My wife is a consultant oncologist, and I just asked whether she was downstairs. So I just called her and asked, if you are big and fat, is there more possibility of having blood cancer? She's a blood cancer consultant. He said
Okay.
no.
Oh,
No,
I mean,
he
that's,
said-
I mean, that's true. I mean, it's
so
a,
yeah, so yeah. But it's good.
That's good to know. No,
I'm so
Ananda,
happy to
yes.
listen to you. No, I'm going to invite you someday to our place, and you can have a good discussion. Yeah, very good, Kshitij. Great,
Thank you.
good seeing you. Yeah,
Next question
Good.
from Vinodananda.
yeah. Pardon?
This is you. Sorry, sir. Okay.
Someone is talking. I think his phone is muted. Maybe
Muted.
Pratyush, you can unmute him.
If somebody wants to say something.
Can you hear me now?
Yes. We hear
Hello.
you. Yes.
Hello.
Yes,
Can
yes,
you hear me?
we can hear you.
Yes.
Yeah. I'm Binodan Mishra, 79 batch. And I'm an engineer by profession, but I did study biology up to 12. One of my biology teacher at BPS Pilani, he made a statement one day that everybody is prone to cancer. And we protested like anything. Then he emphatically said, no, my dear, every human body has got a cancerous cells. It is only matter of time when it gets activated, then you are likely to have a cancer. I don't know how far he was true on what basis he said, but since you are dealing in this subject right now, I want to know your opinion.
This is not exactly true because there is nothing as cancer cell, because cancer is just a growth of undivided cells. It can be in any organ. So, I mean, his point.
No, if I remember correctly, he meant that gene contains that cancerous property.
That's true. I mean, as I said, every cell has, which you call proto-oncogene.
Yeah.
Proto-oncogene are, we call them proto-oncogene because those genes make you predisposed to cancer.
Yeah.
And then there are other genes which kind of tumor suppressor genes. So it's not just because cancer. So those genes, what is cancer? To understand cancer, we need to understand that it's just unregulated, uncontrolled cell division.
Yeah.
Body
So
needs cell division for its growth, for its development, for its sustenance. So there will be genes which we would need for growth, for cell division. Similarly, there would be genes which control cell division and regulate it. So it's so when one of these genes go haywire, for example, if you have genes which you need to control cell division and they should be active only up to a certain part, but they have activated so much that the body doesn't know where to stop. Or the other genes which regulate your cell division, like how many, but they have like completely shut down. They cannot regulate anymore. So that gives the other cell division genes to like complete free hand. I mean, this is how it is. I mean, in a very basic sense.
okay. Okay, thank you.
No worries. Thanks a lot.
Next question. Anybody? I see nobody raising their hands. Nobody has a question. In which case, I will continue my discussion with you afterwards.
Sure.
Because I don't think we should bore people, but extraordinary. Thank you very much for this talk.
Thank you, Pratyush.
And I would like to ask you more questions later.
Sure. Any day, मतलब 1-to-1 कर सकते हैं?
Yes. हमको वही आपसे बहुत कुछ पूछना. नहीं, we have to do this 1-to-1 because if there are people here, no, let's do 1-to-1. It's much better because I started looking at gene replication few years back. आपको शायद ये नाम भी पता होगा. Somebody gave me replication timing data.
Okazaki.
Huh?
Okazaki.
Yeah. Yes, Okazaki. This is the name that I remember. Yes.
Okazaki.
I remember Okazaki name. They have given me data about human cell lines. And it seems that different cell lines, at least to me, what I see, they replicate in different ways. इसके बारे में बाद में बात करेंगे. We have to chat more. Okay. क्योंकि मेरे को बस यही दिख रहा है कि कुछ अलग हो रहा है, डिफरेंस एंड लाइंस डूइंग डिफरेंट थिंग, उसके आगे पीछे मेरे को कुछ और पता नहीं है. बट Okazaki was this name I maybe for us briefly you can explain what Okazaki means. You should
Okazaki
explain
was
it.
just a scientist who figured out how exactly replication happens, and
But these are Okazaki segments, no? That's what we are talking about.
Okazaki fragments, exactly. जैसे
Yes,
कि
yes.
अगर कहीं से रेप्लिकेशन हो रहा है
हाँ.
तो आगे तक बनाएगा, फिर उससे तो छोटे छोटे जो है they are called Okazaki fragments. I mean it's a
This is
cell
what I
replication, transcription, these
yeah,
are like
yeah.
completely independent fields of their own. And I mean I'm also not an expert because bio में आप like cell replication that you see, cell replication itself is regulated and controlled by hundreds of proteins, and they form a complex
Yes, yes.
and it's a field in itself. So I'll also not be an expert. Yeah, as a biology PhD, I can explain the basics, but like very detailed part, maybe I will also not be the best because there are people who
Doesn't matter, but this word I remember, Okazaki segments is what, yes, yes.
people who have done their PhD and doing their postdoc just in DNA replication. So
Yeah, these are heavy, different.
and also, I mean, not just that, replication is totally different. Like the replication in different organisms, different types of cells, meiotic, mitosis.
Yeah.
With every time you have, it's totally different. Meiotic and mitotic cell divisions.
But since we are talking about this, now do you know of this KJMA model, Kolmogorov? This is the cell DNA replication model.
I have heard of it, but I'm not aware because my mathematical modeling courses in it. More of a pure biology courses.
फिर हम आपसे बाद में बात
follow-
करेंगे. We have to talk. We have to infuse mathematics to biology. But this is constrained Gaussian random stochastic process, whether there is a starting point, which is the beginning of where things start replicating and multiples. Lekin, again, later. And with that,
Yes.
thank you, thanks to the participants.
Yeah, thanks a lot. प्रणव दा जो है, प्रणव प्रकाश तो आपके बैच के हैं, आपके सीनियर थे?
एक साल जूनियर हैं, अगर आप सेम प्रणव प्रकाश की बात कर रहे हैं तो.
ऑक्सफोर्ड में अभी हैं वो.
प्रणव प्रकाश.
हाँ.
क्या कर रहे हैं?
He did his PhD in history. उससे पहले सेंट स्टीफेंस.
हाँ, बिल्कुल. वो पल्लव के बैच के हैं, पल्लव के बैच के हैं, हमसे एक साल. हाँ.
तो 2002 हुए ना वो?
हाँ, 2002-12 वो सेंट स्टीफेंस में थे, बिल्कुल.
ओके. हाँ.
इसी समय बहुत ज्यादा लाल सलाम भी थे शायद, if we are talking about. नहीं, वो प्रांशु प्रकाश था, प्रणव नहीं था. प्रणव हमेशा शांत आदमी था. हाँ, प्रांशु प्रकाश बहुत